Hey everybody, this is a little kind of excerpt into what we talked about today, the monohybrid crosses, and an introduction into dihybrid crosses as well. Now a lot of this stuff should be familiar because we did a lot of this in class together. So hopefully this part is a review. Okay, monohybrid crosses. The word mono means one, hybrid means to mix. So what I'm doing is I'm mixing one trait with another one. And we talked about the purple flowers in class with the Mendel and his experiment, so we're going to use these purple flowers. Now initially what happened with Mendel is he took a purple flower and crossed it with a white flower like this, and what he ended up with was 100% of them came out purple. Well, what he didn't know is that they were heterozygous. We can do that now because we can do a Punnett Square to figure that out. So how do we do this? Well, Punnett squares are quite easy. You just draw yourself a little 4x4 square, or actually 2x2, making 4 squares. And then you take the each parent genotype. If you remember, the genotype is the letters. The letters could be big, big, they could be big, small, and they can be small, small. Now in this particular case, this is a flower that is heterozygous for its two traits, or for its trait, and so it is heterozygous, one big, one small. Now this purple flower is also heterozygous, so it is also big and small. So what you do is you take the genotype, the two letters, and you're going to split them in half, right down the middle. Because what happens is during meiosis, which is the process that makes eggs and sperm, they're going to be separated and split into various eggs and sperm, or what we call gametes. Gametes is the nice way of saying eggs and sperm. And so what happens is what we're representing here by these letters is the eggs and the sperm that are going to form when they make gametes. And what can happen is that these two can get together, these two can get together, these two can get together, or these two can get together. So by filling in the boxes by what's above and to the side, it will give me the genotype of what could possibly be. So in this particular case, we are doing two heterozygous individuals. Heterozygous right here means that you have one dominant and one recessive allele. Now sometimes your crosses will have two bigs, and if it's two bigs, well then we call it homozygous dominant. Homozygous means the same, dominant means the two bigs, two of the same. And then you can have two littles, which we call homozygous recessive. So again, homozygous means the same, recessive means of the lowercase letter. So you can do Punnett squares with any of these three and it'll give you all sorts of different outcomes. The outcomes, what we fill in the boxes, can be represented a few different ways. We can represent it with ratios, with fractions, or with percents. The ratios just use the 4 boxes. So in this particular case, crossing two heterozygous individuals, we end up with one box being big, big, two boxes being big, small, and one box being small, small. So I would say it's a 1 to 2 to 1 ratio. Now for the phenotype, what do we see? Well, the big, big and the big, small both make purple flowers. So therefore, 3 of the boxes are purple and 1 box is white. So there's a 3 to 1 ratio. Now for fractions, you can do the same kind of thing. It's just the same as this. Instead of 2 and 1, we just say 1 over 4, 2 over 4, 1 over 4. Same thing over here. 3 out of 4 are purple, 1 out of 4 is white. So ratios work just the same as fractions. Then we have percents, which again is easy. Everything's in multiples of 25. So one of the boxes, 1 out of 4, so 25% are big, big. 2 out of 4, or 50%, are big, small. 1 out of 4 is small, small, 25%. Now 3 out of 4 are purple, so 75% purple, and 1 out of 4 is white, so 25% white. So this is all what we call monohybrid crosses because we're only looking at one trait. I'm looking at just flower color. But if you remember, Mendel did his experiments on many traits, not just purple flowers, but he also looked at the color of the pea pods and the color of the peas and the shape of the peas and so on. So he wanted to know if traits went together. So is it every time I have a purple flower, am I going to always get a yellow pea pod? So he wanted to see if these two were tied together. Were two traits tied together? Like, do all blonde-haired people have blue eyes? Do all red-haired people have freckles? If 100% of the time that were the case, we could say those two traits are tied together. But in his experiments, he found out that most traits are not tied together. And so what he did was something called a dihybrid cross. A dihybrid cross takes a look at two traits at the same time to see how the offspring might show up. Now, in doing his experiments, he came up with this thing called the law of independent assortment. And what that says is that my two traits are not tied together, but they go separately. They go on their own ways. And so if you write this down, genes for different traits will segregate independently during the formation of gametes, so making eggs and sperm. So when an egg and a sperm are being produced, and I've got two different traits, hair color and eye color, they don't go with each other. They're not buddies. They don't go with each other all the time. What ends up happening is that they go independently of each other and do their own thing. And then depending on how you reshuffle those back together will determine what you get. So what I've got over here is two cats. And these cats have some traits, characteristics, which are located right here. So one trait has to do with just the tail, whether they get a short tail or a long tail. Another trait is their coat, whether they have brown or white. So every time I have a short-tailed coat or a short-tailed cat, am I going to get a white coat? Do all long-tailed cats get brown fur? That's what he wanted to see. So again, he performed some experiments with pea plants, not with cats. And what he found out was that they weren't tied together. See, I can have a brown cat with a short tail or a brown cat with a long tail. I can have a white cat with a short tail, or I can have a white cat with a long tail. And how you do this, how we do the dihybrid cross is very similar to doing the Punnett squares with a monohybrid. First thing we need to do is to figure out how to separate everything. So what I'm going to do is I'm going to insert a page here. Okay, and so I've got, let me go back a sec. Okay, so we got big, big, small, 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 big, big. Okay, so I've got a cat who's big, big, small, small, crossed with a cat who's small, small, big, big. So how do I figure out what the eggs and sperm are going to carry? Before it was easy, we just separated them in half. Well, it's kind of true. So these two are going to separate into big S, and the two B's are going to separate into little b, little b, little b. So that means that the eggs are going to carry a big S and a little b. Same thing here. The little s's are going to separate little into the sperm. That's a sperm. And then the big B's are going to separate as well. Big B, big B. Okay. So now what we have to do is figure out how they're going to mix together. So that's where the dihybrid Punnett square comes from. What you're going to do is, just like before, you're going to make a square. But instead of making 4 squares, you're going to add a couple more lines, and you're going to make 16 squares. Oh, that's nice. And then you are going to put whatever the eggs and the sperm are inside, on top of, like we did before. Now, if you notice right here, I've only got two different ones. But what if, let's say our cat was big S, big B, little b? Well, you can see that I can get a big S with a big B, so this big S with this big B, but I can also get this big S with a little b. I can also get a little s with a big B, and I can also get a little s with a little b. So if these two are independent of each other, how do I figure out what the eggs and sperm are going to be? Well, the easiest way to do that is something you've learned in math called FOIL. So let me show you. So S, S, B, B. I'm going to put parentheses around it, kind of like it was math. And what you're going to do, you're going to take your first of each set and write those down. You're going to take the outers of each set and write that down. You're going to take the inners of each set and write that down. And then you're going to take the last of each set and write those down. That tells you what your eggs and sperm are going to be. It's really simple. So on the other one was big, big, small, small. So if I FOILed that, I get big, small from the first. I get from the outers, big, small. From the inners, I get big, small. And from the lasters, I get big, small. So sometimes I get 4 different ones, sometimes I get all the same, sometimes I get 2 different ones. It all depends, but foiling will tell you how many. Okay, so if I foiled out that cat, those 2 cats, I found out that one of them makes all big, small, big, small, big, small, big, small. The other one makes all small, big, small, big, small, big, and small, big. Okay, so what do I do now? Well, I'm going to cross them just like I did before, where you look up and to the side, and whatever it is, you're going to drop it down. Okay? Now, since everything across the top is the same and everything on the side is the same, we know that every box is going to be the same. So that makes it pretty easy. All I need to do is mix 2 together. You want to keep the same type of letter together, and if there's a big and a small, big always goes first. So on this one I have a big S and a little s, so I'm going to write big S, little s. And then I have a small b and a big B, so I'm going to write big B, small b. Okay, and every single one is going to end up the same. So I have big S, little s, big B, little b. Okay, let's go back to the picture. Okay, so do you see that right there? Big S, little s, big B, little b. So what does that mean? Well, they're heterozygous for this trait. So heterozygous means they're going to show the dominant, which is all short tail. You notice every single cat has a short tail. For the B's, big B, little b, they're all heterozygous for the coat color. And heterozygous means you show the dominant trait, so they're all brown. So when these 2 cats have babies, every single one comes out brown and a short tail. Okay, well, what if these 2 mix together? What do I get? So let me go back to that page again. Okay, so I've got a cat who's big, little, and I want to cross it with another cat who's big, little, big, little. Okay, so that's where foil comes in. So I'm going to do my big old square like this. Then, I know, sorry, I should have these pre-made. Be way quicker and much nicer to look at. Okay, so this cat I'm going to foil out. I'm going to start with the first, big, big, then the outers, big, small, inners, small, big, lasters, small, small. Same thing for this one, and I'll get the same thing. Big, 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 small, small, big, small, small. Okay, so what's going to happen? Well, if every one is different, if I had big, 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 small, small, big, small, small, and same thing here, if every single one is different, then there's this glorious, wonderful thing. You can either, A, go ahead and do them all and start tallying them up. Big, 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 small, big, small, big, 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 small, big, small. I could do that for all 16 squares. Bleh, I don't want to do that. Here's the thing. Okay, do you see how we've got these guys right here? Well, if I do all this and fill it all in and count them all up as to what they look like, here's this great thing. 9 of them, 9 of the 16 cats will look like this guy. It'll have both dominant traits. 3 of the cats are going to have this. They're going to have 1 dominant and 1 recessive. So they're going to have a dominant and a recessive. 3 of the cats are going to look like this. They're going to have a recessive and a dominant. And then 1 of the cats is going to look like this. It's going to be recessive, recessive. Okay? Every single time. Only if both sides are different for all 4. Okay? And I always get this wonderful ratio, 9 to 3 to 3 to 1. So let's go back and look. Okay? So 9 will be dominant, dominant. So dominant, dominant, short, brown. 1, 2, 3, 4, 5, 6, 7, 8, 9. There's our short brown. Okay, 3 of them will be dominant, recessive. Dominant, recessive. So 3 are going to be white with short tails. 1, 2, 3. Cool. 3 are going to be recessive and dominant, brown with long tails. 1, 2, 3. And 1 of them will be recessive for both, short and brown. I'm sorry. Long and white, which is this cat right there. And so what's nice about dihybrid crosses, if both parents, and this is a big thing right here, if both parents are heterozygous for both traits, so I take parent 1 and parent 2, and both traits are heterozygous, I get the 9 to 3 to 3 to 1 ratio. Hi. My kids are weird. My husband is dancing outside the window right now. They're nuts. And so this works that way. What if it's not heterozygous for both? Well, then all you have to do is just do the actual Punnett Square. So, 15 minutes. Okay, how about this? We'll practice more tomorrow doing the dihybrid crosses with SpongeBob and explain a little bit more in class. All right, bye guys.